Linkage map of Drosophila chromosome. Linkage map is the genetic map that shows the position of the genetic markers or the genes which are placed on the chromosome in terms of recombinant frequency. Frequency of the offsprings which are produced as a test cross shows if the frequency of the recombinant offsprings is higher, means the genes they are far from each other, while if the frequency of the offsprings which are produced as a cross is less, means the genes or the genetic markers they are closely placed. Greater the distance between two genetic markers or between two genes, means there will be higher the frequency, while on the other hand, if the genes are close to each other, the frequency of the offsprings will be less in case of recombinant. Sturtevant was a scientist who used the recombinant frequencies to map the position of three genes of Drosophila. He used the gene or the genetic marker of body color, eye color, and the wing color gene. With the help of these three genes, he mapped the position of these genes on the chromosome. He designed a test cross, and with the help of test cross, he proved what is the frequency between these three types of genes and how gene can be ordered and placed on the chromosome. How the linkage map of the chromosome of Drosophila was mapped. There were three genes. One gene, that is B gene. B gene was the body color, while CN gene, that was for eye color, while V gene was for the wings. So a chromosome map was designed between these three genes. The recombination frequency between the CN gene and B gene was 9%, while the frequency of recombination between CN gene and VG gene was 9.5%. But on the other hand, the recombination frequency between B and VG gene was 17%. With the help of these three types of the frequencies, linkage map of the chromosome of Drosophila was performed. How we can place or how we can order the position of these genes on the chromosome. If the frequency between B and CN gene is 9.5%, while the CN gene and VG gene is 9.5%, and the frequency between B and VG gene was 17%. It means the CN gene, which was for the eye color, should be placed between the two other genes. So eye color gene should be placed between the two other genes, then a genetic map according to the recombination frequencies was possible. This is the linkage map of Drosophila chromosome 2. In this case, we can see different genes and their locations. This is centimorgan 0 and the length of this chromosome is 104 centimorgan. So the first gene, which is for bristled appendages, is on centimorgan 0. This gene is present on centimorgan 0, while the gray body gene or the black body gene is present at centimorgan 48.5. The red eye gene or the cinnabar eye gene is present on centimorgan 57.5. The normal wings and the vestigial wings, the gene of these two types of wings is present at centimorgan 67, while the red eye or the brown eye gene is present at 104.5 centimorgan. So if we describe the position of the genes in terms of centimorgan or in terms of recombination frequency, such a map is called as linkage map. This is another diagram about the chromosomes and the linkage map of Drosophila chromosomes. Drosophila chromosomes are, this is X chromosome, this is Y chromosome, chromosome 2, chromosome 3, and this one is chromosome 4. So the length of chromosome X is approximately 75 centimorgan, while the length of chromosome 2 is 107, length of chromosome 3 110. So these are the length of the chromosome. And on the other hand, if we see chromosome 4, that is just 1.5 million bases. The length of the chromosome is described in terms of centimorgan, while the physical data or the genes, they can also be described in terms of million bases or megabases. This is the Drosophila chromosome map and the position of different genes on the Drosophila chromosomes. Stultford performed that the genes can be placed on the chromosomes based on the recombination frequencies. He used the units, map unit, to describe these frequencies. One map unit nowadays is equal to 1 centimorgan, approximately 1 centimorgan. And within 1 centimorgan, there can be approximately 1 million nucleotides. But it depends on the position of the chromosome. It can vary. It can be 1 million or it can be sometimes more or less.